Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from the neuroscientists across the schools and departments of our university, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below.
Okay, I'd like to welcome everybody here today to today's Cambridge Neuroscience Interdisciplinary Seminar Series. Today I'm delighted to welcome Dr. Yaara Erez. Yaara was, until very recently, was a member of the MRC Cognition and Brain Sciences unit here at the university and continues to be an affiliated member. But she has just recently returned to her old university, if I'm not mistaken, to Bar Ilan University in Israel, to the Faculty of Engineering there. But Yaara is going to speak to us about the work she conducted at Cambridge here. She was here as a postdoc with John Duncan before she started Royal Society Dorothy Hodgkin Fellowship, if I'm correct. And so she's going to tell us about her work here and I guess all her plans for the future for her new research group. So over to you, Yaara, and we're really looking forward to your talk.
Thank you very much, Dervila, for the kind invitation and introduction. As you said, I just opened my lab at Bar Ilan University. And today I'm going to talk about cross-modality imaging of the neural systems that support executive function. And all of this work was done back in Cambridge, and parts of it in collaboration with the neurosurgery department in Cambridge University Hospital and the psychiatry department. And I'll start with what I want you to take from this talk. 3 points. The first one is that research at the individual participant level is essential. We normally look at group averages, but we should really start looking more closely at individuals. And this is particularly important when we work with patients. The second point is that neuroimaging techniques or modalities can and probably should be combined. We have very good tools to work with each of them separately. And we are now starting to move more and more towards integrating data or findings or both across techniques. And the third point relates to the relationship between basic science and clinical applications. So we normally think about it as a unidirectional path. We have tools and knowledge that we gain in basic science, and then we apply it in clinical settings. But actually, this is a bidirectional path because we can also use the data and the findings that we obtain from clinical research to learn more about basic neural mechanisms. So I'm going to address these points. And when we want to better understand the link between the brain and behavior, we can look at brain networks, areas that work together, and their functionality. And what I'm showing here is data from a few years ago from the Dosenbach Club. This is the Midnight Scan Club first paper. And this is resting state fMRI data, and it shows different networks, areas that are functionally connected in different colors. I highlighted just a few here at the back, visual regions, auditory regions here in light purple, the hand and face motor and sensory areas, and there are many other functional networks. And when we want to understand these networks, well, we have a lot of challenges, but I'm going to highlight just a couple for the purpose of this talk. The first one is mapping or identifying networks in individuals. This is data from the same paper as the previous slide, but this time I'm showing data from 4 different participants, and you can see how much more detailed the maps are. Normally, we average across a group of the participants, average the data, and that's very useful because then we can get rid of the noise and we are left with just the signal. But at the same time, we actually lose a lot of details. And if we want to understand and get a picture of brain function in greater detail, then we should also look at individuals. And the second challenge is related to the integration of neural data across the different imaging modalities. We have a variety of modalities. I'm only showing a few here. Very good tools and analysis methods for working with each of them, but it's much more challenging to integrate across them. It's important to do that because each technique measures something slightly different at a different scale, at a different resolution, with different advantages and disadvantages. And if we can integrate across them, then we can, for example, look at data across a different scale and different resolutions. I'm going to show you how we address each of these challenges, and I'm going to focus on executive control functions, which have been the focus of my research in the last few years. You can see in this picture of a kitchen service, the people working, preparing the meal, and they need to do a lot of things. They need to pay attention to a lot of details and also discard what they don't need, the irrelevant ones. They need to plan their actions ahead. They need to solve problems when they arise. They need to attend to different tasks and switch between them very quickly. And this demonstrates this family or collection of functions that include attention, planning, problem solving, task switching, and so on. And we use all of these all the time in our daily life, whenever we prepare a meal, like here in the picture, but also when in everything that we do, when we pack a bag in the morning to go out of the house, when we plan our holiday, when we host a party, pretty much in everything that we do. So these people in the kitchen, even if they see perfectly well, even if their hands move perfectly well, if their executive control functions don't work perfectly well, then they can't make this service successful. At the neural level, we know that a collection of regions in the frontal and parietal cortex are involved in executive control. So you can see here in the frontal cortex, a band along the inferior frontal gyrus, an area here just anteriorly to FEF. This is fMRI data, by the way. An area here just anterior at the anterior insula, and on the medial surface, the pre-SMA, the intraoperative sulcus. This is a visual component that is not considered as part of the network, but we see it here because visual tasks were used in this study by Ev Fedorenko. And this is a bilateral network. I'm showing only one hemisphere here, but it's a bilateral network. It has been termed by John Duncan the multiple demand network to reflect its domain general nature. But it's also termed the control network or the frontoparietal network, and all of these names refer pretty much to the same areas and network of regions. We can quite easily see this network in fMRI. This is actually a very consistent and robust evidence across many different studies. When we ask participants to do a task and then ask them to do the same task but a harder version with an increased cognitive demand, we can see the recruitment of this network. And it has been previously shown across many different studies to be involved in a range of executive control functions, such as attention, reasoning, problem solving, and so on. More recently, Muataz Assam and colleagues provided a more anatomically detailed description of this network and also divided it into core and an extended network, but this is work with the HCP data. But again, it's the same network that is involved in executive control. And with this in mind, I'm going to talk today about two main projects. In the first one, we looked at how we can localize and how well we can localize this network in individuals. And this is an fMRI study with healthy participants. And in the second part, I'm going to talk about our work with patients when we mapped executive control regions with ECOG, with electrodes that are placed directly on the surface of the brain during awake surgeries. And I will explain exactly what we try to do there later. So I'll start with fMRI in healthy participants. This is a study led by my former students, Niha and Flurje. And when we studied the network, the frontoparietal network with fMRI, we wanted to understand better how this works, how the different regions contribute to the different functions, what kind of information is represented, for example, as we can measure with MVPA. Across this network, what is the differential contribution of perhaps subnetworks? And we realized that in many cases, the effects that we see are actually very weak. So we were thinking about it, and we thought perhaps we're just not looking exactly in the right places. And this is based on work of others that already started looking at this network as defined in individuals. And we wanted to look at that and understand how well can we localize this network and systematically assess that. So we used 3 different tasks that were previously used in the literature: a spatial working memory task, a verbal working memory task, and a Stroop task. All look at different aspects of executive control. And for each of the tasks, we had an easy task, an easy condition, and a harder condition. And we can look at the change in activity between those conditions with increased cognitive demand. And this is how the group average map looks like. This is a good sanity check. We can see this typical pattern of activity for the frontoparietal network with this frontal patch and the IPS visual components, because it was a visual task, and the pre-SMA over here. But what we were really interested in is how it looks like in individuals. So what I'm showing you here is maps from 6 different participants from one of the tasks. And I hope that you can see that they all show, again, this typical pattern of the network in the frontal cortex and the parietal patch. But what you could also appreciate is the large variability across participants. I highlighted a couple of places, but you can see that the differences are much more than just these 2 places. And we wanted to quantify them. So what I'm showing you here is not the standard fMRI activity map. This is a probability map. So the color of each voxel shows the number or proportion of participants that had significant change in activity with increased cognitive demand. So the yellow parts is where areas where most participants had such change in activity, and the reddish or darker parts are where a smaller proportion of participants had such a change. And we can see these hot spots in yellow or brighter colors with this typical pattern of the frontoparietal network. But we can also see how widespread this is, reflecting this variability across participants. What we asked next is so we can see different patterns of activity for the different participants, but how reliable are they? So for each participant, we repeated the same task twice, and we could look at the similarity of the pattern using a correlation across voxels, so simply correlating activity in each of the runs for each of the tasks separately for each participant. And these would be the darker bars here. These are reliabilities for the first task, second, and third. And you can see how high these reliabilities are. 0.7 and 0.8 is really high for fMRI data. And what this means is that whenever participants repeat the same task twice, this pretty much recruits the same areas in both repetitions. we also correlated activity across voxels between participants. So a pattern of activity for one task for one participant with the same task but a different participant, and all possible combinations of pairs of participants. And these are the lighter bars. And you can see how this correlation drops, reflecting this variability across participants. So patterns of activity are reliable in individuals and variable across individuals for this network. What we wanted to ask next is whether these areas that we just saw that are so reliable, whether they reflect the executive control network more generally. So perhaps it's not surprising that if we repeat the same task twice, we see the same regions, but does it mean that they are more generally related to executive functions? And this is what we looked at next. We used another task that each participant did in the scanner, a fourth independent criterion task. This was a task switching task that was also previously used in the literature. So we knew what to expect. Activity is expected to be higher for switch compared to state trials. And we used each of the areas that we identified earlier in each individual with each of the tasks as an individually defined region. So each of the tasks was used as a localizer based on the approach that Ev Fedorenko used about 10 years ago, first in the language system and later on in other systems. As a start, we looked, instead of the localized regions, we looked at what happens when we define the same regions for all participants using a group template. So this is a very common practice in fMRI studies. We look at specific regions of interest, and what most studies do, they define exactly the same regions for all participants. But the good news is that we were able to replicate the effect that was previously described in the literature, so that was nice. And when we looked at the individually defined regions, not only we could see this effect again, but the effects were actually much larger. So from that, we can conclude that the individually localized regions are more generally related to executive control, and also we can achieve larger sensitivity in these individually localized regions. So we looked at several other things in this paper, which I'm not going to talk about, but I just want to mention that we also use this approach with the individually localized regions in other studies and other fMRI data analysis approaches, for example, with MVPA, when we looked at pattern across voxels instead of average activity as before. And this was an attention task. And we were able to, using MVPA, to look at the information that was represented across this network. And we were able to identify representation of relevant information, but not irrelevant one. And this is another core aspect, this representation of only extraction of only relevant information and discarding of the irrelevant one. This is another core aspect of executive function. So this approach looks very useful, but it's particularly useful for work with patients. Just to summarize this part, this network is unique in individuals. We can reliably localize it. And why is this so useful for work with patients? Because when we treat patients, we want to treat the patient that sits in front of us and not the average hypothetical non-existing patient. And this is why it's so important to look at maps and identify them in the best possible way in individuals and not as an average. And that leads me to the second part of the talk and our work with patients with brain tumors. As I mentioned at the start, this is a large collaborative project with people from Cambridge University Hospital, the neurosurgery department, and also the psychiatry department. And there are many different types of brain tumors. What the patients that we worked with in this project were diagnosed with a diffuse low-grade glioma. You can see here in this MRI scan of one of the patients, this darker gray blob here at the top. This is the frontal cortex. Low-grade gliomas account for about 15% of all primary brain tumors, tumors that arise in the brain. The average age of diagnosis is rather young, normally between 30 and 45 years old. And the average survival after diagnosis is rather long, 10 to 15 years. On average, people can live with this disease for 20 years or even more. What is typical for these tumors is that they are slow growing, so they are not aggressive, and they were probably present in the brain of the patient for months and even sometimes years before diagnosis. And that means that they can also live with the tumor for many more years. But the bad news is that it spreads into the nearby healthy tissue. So there is no clear boundary between the tumor and the healthy tissue around it. In many cases, the first line of treatment for these tumors is a surgical procedure for the removal of the tumor. And during this procedure, what neurosurgeons need to do is to optimize a very fine balance between removing as much of the tumor on one hand and preserving function on the other hand. Because the tumor infiltrates the nearby tissue, it's very important to map the essential functions around the tumor so we don't damage any essential functions. And patients can have good quality of life following surgery, so they can perhaps go back to their normal work and just have normal life like we all have. And the way this is done in many cases is using an awake surgical procedure and functional mapping with stimulation. So after the skull has been opened during the surgery, the patient is being awakened, and then they are asked to do different cognitive tasks, simple tasks like counting or looking at pictures and saying what they see. And while they do that, the neurosurgeon applies brief pulses of electrical stimulation on different parts of the brain. And the idea is that if stimulation leads to impairment of function, reversible impairment of function, for example, the patient stopped talking, then this means that this area may be critical for this function, in this case, speech production. And a neurosurgeon might decide not to take this part out, even if from an oncological point of view, it may have been better to take it out. And this functional mapping procedure with stimulation is now done in many centers around the world, especially for motor movement and some aspects of language. It's very coarse mapping, but it's better than nothing. And there is effort in the field to expand this to other functions as well. For executive control functions, such mapping is quite challenging for many reasons, but particularly because of the complex nature of these functions and because of the distributed network that supports them. And there are only a handful of studies in the literature that address this using stimulation, usually focusing on one aspect of executive control. In this case, is the Stroop task that looks at inhibitory control. And our idea was to use ECOG, recording of brain activity, instead of stimulation, to identify the executive control network. And instead of tapping on just one aspect of executive control, we want to recruit the entire network with the idea of identifying a neural signature of control regions and eventually guide stimulation. So a neurosurgeon applies stimulation on different parts of the brain, and it takes time. And we want to make this procedure more efficient. If we can guide them to the right areas, then this could be very helpful and makes the procedure shorter and safer and so on. So that's what we are trying to do. And Our research has been covered by the BBC a few years ago. This is Ben. He has been diagnosed with a brain tumor and has kindly agreed to not only take part in our research, but also participate in this film by the BBC and have his surgery filmed. And I'm going to show you now just a short part of this movie. You can see the full one on YouTube, and there is a link from my website. And I want to highlight two things about this short part. In the first part, you will see the neurosurgeon applying the stimulation, and you will see how Ben stops talking. In the second part, you will see Ben while he's participating in our research and doing one of the tasks.
A few months later, surgeon Thomas Santarius is removing it, while Ben is still awake.
Okay, take a break a little bit. Can I have a 2.5?
We were even allowed to talk to him.
Hi, Richard.
I know these are very strange
It's Richard,
circumstances.
I've got it right. I'm still working. Yeah,
You've been doing amazingly well, and you've still kept your sense of humour.
yeah. Try to kept to laugh, eh?
Using an electric current, Thomas could switch sections of the brain off. Listen to this.
Can you count for me again?
1, 2, 3, 4.
The fact that Ben stops counting means it's a part affecting speech, so Thomas won't remove it.
We'll start now.
But it's not the awake part that makes this operation special.
A1, B2, C3.
Yaara Erez isn't a doctor, she's a neuroscientist.
F6.
This test is hard enough when you're not in the middle of surgery. You try doing it.
8, 5, 9.
By forcing Ben's brain to multitask, scientists are creating a much more detailed map of its functions for a new device that will eventually help surgeons decide what to take out and what to leave behind.
So I hope you could get a flavor how this looks like in the operating theater. And I'm now going to tell you a bit in more detail what we are actually doing. And this study was conducted together with my former student Muataz. And this is how the intraoperative setup looks like. The patient is lying doing the surgery. Electrodes are temporarily placed on the brain. They are removed after we finish the research part. They are connected to the acquisition system that records brain activity. We use the neuronavigation system that we have in the operating room with this probe to identify the location of where the electrodes are placed. And we record brain activity while the patient is doing cognitive tasks. The aim is to provide neurosurgeons with feedback in real time during the surgery. But as a start to establish this approach and measure, we analyze the data offline. And everything that I'm going to show you today is from such offline analysis. The main benefit of the ECOG signal is that it has both high temporal and high spatial resolution that we can't achieve with non-invasive techniques like fMRI or EEG. What kind of tasks did the patients do? So as I mentioned earlier, with fMRI, we can identify the executive control network with an increased cognitive demand manipulation. So we use the same idea here. Each patient performed 2 tasks, an easy and a harder one. The easy one was simply counting from 1 to 20, and the harder one was counting while switching between letters and numbers, 1A, 2B, 3C. This becomes hard very quickly. And we can look at the change in activity between these 2 conditions. And what we're looking for is a neural signature of executive function, something that will tell us these are regions that are related to executive function. And we decided to focus on a specific measure in the signal, the high gamma power between 70 and 250 hertz. And we thought that this measure would be useful for several reasons. It has been previously associated with cognitive processing. It has been linked to both the fMRI signal and spiking activity, and it has been also used in the brain-computer interface. And I should also say that the high gamma activity in the high gamma range cannot be identified in EEG. We can only see it in ECOG. Based on these previous studies, what we expected to see is increase in power with increased cognitive demand, increased in power in the high gamma range. What I'm showing you here is examples from 3 patients. This is the frontal cortex, and red dots or circles, each is an electrode. We used strips with 4 electrodes each, and after bipolar re-referencing, we are left with 3 electrodes per strip. And you can see the color coding. Red means increase in power in the high gamma range with increased cognitive demand. Blue means decrease. And you can see that in the frontal cortex, in areas that we will expect to see activity that is related to executive control, we see this expected increase in the high gamma range. What we did next is to look at the same measure, but this time with a larger cohort. So we recruited 22 patients, and we have complete data that we could use from 13 out of these 22 patients. I'm showing you here all the electrodes superimposed on the left hemisphere, but we had electrodes from both the right and the left. Each dot is 1 electrode, and you can see this red blob of activity in the frontal cortex increase in the high gamma with increased cognitive demand, as we would expect. Expanding these findings from previous reports that looked at it in other domains, such as the motor system and the visual system. We also looked at activity in each single electrode, because ultimately that's what we want to achieve. And we used a statistical test that we developed that preserves the temporal correlations in the signal. And you can see again this collection of electrodes in the frontal cortex with increased power in the high gamma range with increased cognitive demand. We also looked at another frequency band, the beta band, which has been previously associated with an attentive state. This time, higher attentive state is associated with decrease in power in the beta frequency band. And this is what we found in line with previous studies. But what was interesting was that the increases in the high gamma were rather focal in the frontal cortex, while the decreases in the beta were much more widespread. And the next step was to link it more formally to executive control regions, because the frontal cortex, not all of it is part of the frontoparietal network. So for that, we used the fMRI data, and for each electrode, we determined whether it lies within or outside the frontoparietal network. For the fMRI data, we used the resting state data from the Yaara and colleagues 2011 paper and the frontoparietal network as presented there. And then we looked at several measures. For example, we looked at the proportion of electrodes that showed significant change in activity. Above the 0 line is the proportion that showed power increases. Below the 0 line, those that showed power decreases. And we can see for the high gamma that increases were much more likely within the network in the yellow bar compared to outside the network in the green bar. And for the beta range, decreases were likely, equally likely both within and outside the network. And this provides evidence for these increases or this neural signature in the gamma power as actually related to executive control regions. We also looked, I will only show you one more measure, overall power change across all electrodes. The picture is very similar. Increases in the power, in the gamma range within the network, decreases in the beta, both within and outside the network. And this provides this evidence for the high gamma power as a good measure. And I want to also highlight that there is converging evidence here across ECOG, what we measured, and the fMRI resting state canonical network that was presented in the Yale paper. So far, what I showed you is data from electrodes that were placed on healthy brain tissue further away from the tumor. But what Ayan was interested in was what happens on the tumor itself. We have evidence from the last couple of years or so, really emerging evidence that that tumor itself is not completely dysfunctional, but rather integrates within local circuits. Most of the evidence, it's very sparse, mostly from the more cellular level. And what Ayan did, he looked at the electrodes that we had that were placed on the tumor. This is still work in progress, and I'm only showing you here a couple of examples from two patients. Two electrodes were both in the frontal cortex on the tumor. You can see this blurred area here indicating the tumor. And they both showed, surprisingly, a change in activity in the high gamma power with increased cognitive demand. So it's the same color scale as before, and you can see this red dot. But what Ayan also wanted to know is how these local activity related to whole brain network. So for each patient, we also had collected neuroimaging data from before the surgery. And Ayan looked at resting state fMRI data and used the location of each of these electrodes individually in each participant as a seed, and looked at the whole brain networks that were functionally connected or with correlated activity across the whole brain. And you can see that this is not nothing. We can clearly see some functional connectivity here with the area that is on the tumor. And he also looked at it more closely. And this is part of the network that is related to executive control and attention. And this is statistically, also statistically overrepresented. So more than what we would expect by chance. So this is not just nonsense. So this is initial evidence for this integration of the tumor within both local and large-scale networks. And it demonstrates the power of what we can achieve when we look at data across imaging modalities. We had several data, quite a lot of data that we collected for each participant, including DTI, resting state fMRI, both before and after the surgery, including neurocognitive assessments. And for those who are interested, we have a series of papers that are already published from this data set. And with that, I would like to summarize. In the first part, I showed you that the executive control network is unique in individuals and can be reliably localized. And in the second part, I showed you that we can identify a neural signature of executive control regions with ECOG, and what we can achieve with cross-modality data integration. And I'm going back to the take-home messages, and I hope that I was able to demonstrate and convince you that these 3 points are important, and we should strive to go more and more towards this direction. I would like to thank all of my collaborators back in Cambridge. Not all of them are in Cambridge anymore, but some. The funders, a special thank you to the patients who agreed to take part in this research. Thank you for your attention, and I'm happy to take questions.
Thank you, Yaara, for that wonderful talk, and wishing you every success in your new position. Congratulations. Join us next time when we welcome Dr. Petra Vertes. Petra leads the Systems and Computational Neuroscience group in the Department of Psychiatry, where she is an assistant professor, and she is also a fellow at the Alan Turing Institute and a visiting researcher at the MRC Laboratory of Molecular Biology. Her research applies tools from physics, engineering, and network science to fundamental problems in neuroscience and mental health. In particular, she is interested in the structure-function relationship in brain networks, from the microscopic scale of neurons to the large-scale connectivity of brain regions in both health and disease. She will be speaking to us on network science and network medicine, new strategies for understanding and treating the biological basis of mental health. For more info on this seminar series and all things neuro-related in Cambridge, follow us on Twitter @CamNeuro and follow the links below. See you next time.